All right, folks, 12:30, let's start. So, just a few things that I'm not going to talk about the exam one yet because I just posted, I just get an email about going through the exam and everything. Remember that the exam will be open for you review several days. So it's not that I just open it for an hour or two. So you see the correct answer and everything. And after that, if you need an explanation of the questions, next week, once these are all genuine documented makeups. So once they're done, if you have a question regarding explanation and nothing else, that you are just comfortable that what you were studying, that's okay. Then you can just ask for schedule an office hour, and that's fine. But if you just want a strategy that you want to improve, need some tips, then just remember that I posted the announcement that in the study technique that you will see that I posted it earlier. There is an Excel file there. The first one tells you that a particular question that one you missed. So just choose one. And the second actually tells you about the study technique. So fill up that and then mail it about 24 hours before you schedule an appointment so that I can take a look at that. And then we come up with the strategy. Because if I do not know quantitatively what you have done, then we cannot. Because if you just come to my lab, it's like I have everything that what you have told me, but still I did not do well. That everything may not be measured. So this is essentially evidence-based teaching. So I just wanted to make sure that you just do that. This will help you out a lot. Just give me 1 sec. All right. So yeah. So again, the important thing is that we just immediately go dive into the next one, and that's where the intensity of that. And if you just take a look at the class, that you can see that after the exam, it's pretty empty, and that's where the thing that people cannot keep up with the intensity. And some of them, maybe up because they do have a background, but that is what it is. Essentially, it's like as soon as you walked out of the exam one, you saw that unit 2 is free at that time. So that's essentially what it is. So it's stiff, so you already know that what is the kind of pattern that what you're asking for. So this will tell a little bit about the study every day. All right, so what we're talking about is a cellular endomembrane, and essentially that you all know that we are talking about essentially we talked about the plasma membrane, but what we essentially mean that these cellular endomembrane are essentially all the membranes that are present in all the membrane bound organelles. That is essentially your nucleus, your endoplasmic reticulum, your Golgi, lysosome, peroxisome, and the plasma membrane. Ribosomes and centromeres, centrosomes, they are not membrane bound, but pretty much all other things are membrane bound, and mitochondria as well, protoplastic plant cells, all membrane bound. And what we are going to see here, that giving a snapshot of these proteins that are synthesized in the cell, how they are transported in different parts of the cell. Now it is almost impossible to cover that whole thing, so we're going to see that we are essentially going to add a certain pathway that will cover your ER, lysosome, and the plasma membrane, and outside. What we will not study in detail, which you can all study on your own is needed, is targeting into the cytosol, essentially the nucleus, as well as chloroplast and mitochondria, which essentially mimics the same kind of strategies, but just different set of proteins. So the picture that you are looking at right here is essentially the nucleus, and this is the blue one is the nucleus, and this is the ER, easily the largest organelle, and these are some of the mitochondria that what you are seeing. And here you are seeing that this is the surface of the membrane, and these are some of the proteins associated with a coat protein, which is called a clasp ring, which is essentially docking onto the membrane, and it is pinching off a part of the membrane. Think about that. You just have a balloon, which is not inflated that much, and like it is inflated, but it is not that you can just grab with your fingers and take a part of it within your palm. That's exactly how they are pinching on the vesicles, and this is a formed vesicles. Once this is formed, this whole structure will essentially disassemble. There are some proteins that will deliver them to the target. So the first part is essentially that what we are talking about, targeting proteins to and across the endoplasmic reticular membranes. So the first thing is that this is entire overview of the eukaryotic protein sorting. And again, there are other courses that tells you about the DNA replication, the transcription, translation, and this is beyond the scope. And there are other courses in Clemson that actually teaches that. That's why I just do not do that. Essentially, just remember that your DNA in the nucleus replicates and equally divided to a new cell. The DNA is transcribed. Essentially, the process is transcription into the RNAs, different types of RNAs, ribosomal RNA, make these ribosomes along with the proteins. Then you have the transfer RNAs that during the protein synthesis brings the amino acids to be added one after another. These are the dots of these amino acids in the protein chain. And the mRNA is the red one, which is the message that is embedded for a protein in the DNA, and that is read by the ribosome to make the protein. So just make sure that you're not familiar with these terms, transcription, translation. Just do your, this is your prereq that's in there. Just do your homework about this so that you're clear about the transcription, translation. We're not going to talk about that. Just some basic preliminary understanding is good enough. So essentially what you're seeing that from the nucleus, all these RNAs that are transcribed are transported into the cytosol. They are synthesized usually in the nucleus region, comes to the nuclear pore, and now they are doing the translation. And mRNA is read. tRNA brings in each of these 20 amino acids that is added one after another, and as per the codons that you have in the mRNA. And this is essentially the growing polypeptide chain. So now, this is what we call as a polyribosome. At the same time, just to increase the efficiency, a single mRNA will start with the start codon, is bound by several ribosomes, and it increases the chances of the production of protein efficiency. Just remember that we talked about the B cells. Per second, it can actually secrete out like 10,000 plus antibody molecules per second. So you can just well imagine that how fast that protein synthesis can actually go on. So now where should these proteins go? That is the most important part, and that's what we are talking about in here in bits of protein targeting. So if this protein that is coming out from the translating ribosome, this is basically a picture of a translating ribosome. You have a smaller subunit, larger subunit, and the mRNA. If this doesn't have any kind of address embedded in there, the destination of that is a cytosolic protein. Very important, just remember that. If you do not have any destination or any address embedded into a protein that is coming out of the ribosome, it remains in the cytosol. It's not targeted anywhere. It's a soluble protein targeted. Now, if you look at on this side, we'll come back to here later. Right here, you have a target sequence. See that the pink one is a target sequence, which is essentially a stretch of amino acid that can be recognized by certain proteins. And depending upon that targeting sequence, they can be targeted in the nucleus, they can go to peroxisome, they can go to chloroplast or mitochondria. All have different addresses. This is the same thing that the mailman delivers your mail. All have different addresses, and these addresses are essentially amino acid sequence, which are in the targeting sequence. Now, these targeting sequence could be at the terminal, it could be in the middle, it could be at the N terminal, it could be at the middle, and it could be the C terminal. You have all different things, but you have to have an address to get them targeted. Without that, it will remain as the cytosolic protein. Now, we are not going to talk about these things. We are interested in this, which we call it as a secretory pathway. And the name is kind of like a little bit misnomer, that the proteins which are synthesized is not, some of them are secreted outside the cell, but not all of them. So that's why it's kind of a misnomer, so know that. So what exactly is happening? That all of these proteins that are going to go through the secretory pathway must have a sequence and have a sequence which is amino acid sequence, and what we call as an endoplasmic reticulum signal sequence. Again, this could be at the N terminal, which is shown here as such. It could be in the middle, it could be even at the C terminal. Really doesn't matter. And also, it gives you another important thing, that there are other other signals embedded in there, which tells you that whether this protein should go onto the membrane or it should remain soluble. So you can see that this ribosome is coming onto the endoplasmic reticulum. This is your endoplasmic reticulum. And now there are two proteins that what you are seeing. There is one protein. This is embedded into the membrane. So the gray part is the membrane of the ER. And then you have this protein, which is in a soluble form. And these are a membrane protein. These are the membrane protein. And you can see that this signal is actually cleaved from these proteins. So some of the proteins which does have an N-terminal signal that's cleaved. But essentially, all the protein that comes to be manufactured on the membrane of the endoplasmic reticulum does have just a definite fate. They are not going to go into the cytosol, not chloroplast, mitochondria, peroxisome, or nucleus, but they can remain as a soluble, remember that, this very carefully. They can remain as a soluble resident protein of the ER, or they could be a protein of the membrane of the endoplasmic reticulum. These are resident proteins. Then, some of the protein can go into the Golgi. They can be the enzymes of the Golgi, so they can remain in the Golgi, or they can remain in the membrane of the Golgi. This is your second target. Then, some of them from the Golgi can go into the lysosome. And even in the lysosome, some can be present as membrane proteins on the lysosomal membrane, or some of them could be in the lumen of the lysosome as soluble protein. Then again, you have the third set of protein that are also going onto the vesicle and goes onto the plasma membrane. It can become a plasma membrane protein, and what the secretory pathway says, and some of them can be secreted outside. So why we are choosing that? Because it tells you that you have a 1, 2, 3, 4, and 5 destination is covered in the secretory pathway. And essentially the mechanism is very similar to this. These are just different protein, different recognition particles, and different target organs. But essentially you will know it all, that how it goes. So again, repeating that, you know that for each of these organelles, you have some resident proteins which are on the membrane of those organelles, or enzymes as a soluble protein in those organelles, and it can go to the lysosome, plasma membrane. But remember that not peroxisome. Peroxisome have different hydrolytic sequence. Only lysosome and plasma membrane, and secreted outside. All right, so just remember that the take-home message, that without any targeting sequence, it remains in the cytosol. That's one of the take-home messages from here. After that, these are some of the self-explanatory slide about just the structure of ER. There is nothing, should not stop on these things. You can just take a look at these. Rough endoplasmic reticulum are embedded with the ribosomes. That's what we are going to see essentially. Smooth does have different lipid biosynthesis, carbohydrate modifications, and synthesis that we are not going to talk about. We are mainly talking about the rough endoplasmic reticulum. And they are a continuum. There is no separate areas. The smooth endoplasmic reticulum is just certain areas where you do not have these embedded ribosomes. That's the only different thing here. All right, so after that, we already talked about that how we isolated the different organelles. You already saw that you essentially break up the cells, and after that, you just homogenize the cells, and then you do initially a velocity, differential velocity centrifugation, then you do density centrifugation, and you collect your sample, which is rich in one of the organelles. So you can well imagine that this organelle is in the entire part of the cell is covered with this endoplasmic reticulum. So you cannot isolate the intact endoplasmic reticulum. So there is a particular way that what you do, that when you break the membrane, essentially, the membrane is made up of this phospholipid bilayer, and you already now know that the phospholipids essentially will just automatically form these micelles or liposome-like structures instantly in an aqueous solution, because all they want is that the outer part, which is the hydrophilic, that must be facing the water, and the hydrophobic core must be towards the inside sandwich area. So what happens, and this is the unique part of the phenomenon, this large rough endoplasmic reticulum, when you homogenize, you just break them apart, but they instantly enclose the material inside and just becomes tiny bubbles. Just think about that. You are taking like, it's not exactly apples to apples, but similar mechanism. You take a beaker, put some oil in there, and you shake that, and immediately the oil doesn't mix with water, but it just breaks up in tiny oils, and then they will come together. So those tiny droplets of oil that separates after the mixing is essentially representing that. The unique part is that this happens so quickly. The material or the enzymes that are present in the inside the ER, they are never exposed to the solution or the homogenizing solution that you are extracting with. So essentially, the interior of this is biochemically equivalent to the ER, and we call these as microsomes. Microsomes are not present in a cell normally. These microsomes actually are derived when you actually isolate the ER. So the isolated ER essentially exists in a microsome format. And if it's a rough endoplasmic reticulum, they are embedded with the ribosome. And if they're smooth, they will be smooth. And now, so these are some of the pictures of the microsomes that you can see, the transmission electron micrograph. And this is how you can isolate the two so that you have a pure sample and you can do experiments with that. So as we know that you just do rough and smooth mixture, and you run them, of course, essentially you just first precipitate all the membrane into membrane structure. After that, you just load them onto the sucrose gradient, and now you can see that they are separated because these are embedded with the ribosome, so they are heavier, so they moved down the concentration gradient from, these are lighter, so they are floating out. And then you actually isolate these microsomal fraction. We have talked about that. The enzyme composition of the smooth and the rough absolutely varies. So there are totally different enzyme compositions. So when this thing's coming out from the, you puncture the bottom of these tubes and collect the samples, and you use a marker enzyme for analysis, and you see a peak, you just pull those samples, which will contain your microsomes. So we are going to see that how the proteins are manufactured on the membrane of the microsomes with the help of the ribosome, and some experimental techniques as well. So the first thing that came into the idea that this was an idea that how essentially the proteins which are synthesized through this secretory pathway to the ER, how it goes to the ER, that was the thing. And it was hypothesized that there must be some kind of signal that is present in the protein. And that used to be called the signal hypothesis. And 1970s, Gunther Blohm actually discovered that what is the signal by doing some elegant experiments, and he was awarded with the Nobel Prize with that. So now it is no more a hypothesis. It is known that what is the procedure, what is the molecular mechanism, and how the proteins are essentially targeted onto the ER membrane. So what happens that we are going to see that this is a very generalized picture. So this is a cutout of the ER. And here you are looking at the ribosomes, and the ribosomes actually capture the mRNA and is brought onto the ER by a certain particle. So these are other molecules. We're going to see that. And then they dock onto a structure which we are going to see is called a translocon. This would be a new word that you may not have seen before. It's called a translocon. It's a barrel-like protein that spans the ER membrane. And once the ribosome actually docks in here, it just insert the growing polypeptide chain into the translocon. And the translocon does have a repetition sequence for certain ER sequences. Without that, it will not allow any protein to enter into the ER, endoplasmic reticulum. And what you can see that along with that, there is also another protein called signal peptidase. That signal peptidase will cleave that part. So here, this red part is the signal, and the green part is the mature protein. The signal is essentially cleaved, and this protein is released into the lumen of the ER. Now, this is a soluble protein. Remember that. This is a soluble protein. Where could be the destination? Depending upon that, it can remain in the ER, can go to the Golgi, go to the lysosome, or secreted outside. But this is not a membrane protein. We are going to see that the membrane protein will be inserted into the membrane, and it still could be in the ER, Golgi, lysosomal membrane, or plasma membrane, but will not be secreted. So either in the lumen or membrane associated. So let's take a look at how this signal is essentially recognized. So this is how, with this elegant experiment, it was done. So just know that how we do this polyacromide gel electrophoresis with the detergent, like SDS, we call this SDS page. So if not, just do a one-line Google search like SDS page, PAGE, it's SDS, sodium glutathione sulfate, polyacromide gel electrophoresis. So essentially what happens that this is a gel which runs in presence of this sodium glutathione sulfate, which is a detergent. All the proteins at the top, and then you just electrophorese this in a buffer. This is the negative charged end, this is the positively charged end. Most of these proteins are negatively charged, so they are going to migrate, bless you, they are going to migrate towards the plus end. And what happens? This polyacromide is essentially a seed-like particle. So the smaller molecular weights will move faster and go farther. Larger molecular weight will be towards the top. So you can just imagine if you see a smaller band like this, or a band like this, or this, molecular weight of this will be less compared to that. That is your take-home message. Just take a look at that, how this is essentially read. So what they have actually seen, that this is an in vitro experiment, in vitro translation, and you can do this. You can just take a ribosome, you can take the mRNA, you add the tRNAs and all of the essential amino acids in there, and then you can do an in vitro translation of the protein. And this can be nowadays, it's kit at that time, it was just being developed. But when this was done, it was done in the presence of microsome and absence of the microsome. So global in that in vitro translational mixture where you have the mRNA, ribosome, tRNAs, and everything, they just added the microsomes or did not add the microsome. And after that, they essentially loaded the samples without the microsome and with microsome, and they see essentially there is a difference in the size of these. So technically, the question is that how will you essentially see? Because when you are just adding these microsomes and all these kind of things, there is a large number of, this is just, if you are just interested in that extra mile, this is not needed for your exam, is that when you have a mixture of all of these, how would you just know that there is just only one band of this? Because this contains zillion of proteins and it should be like a black band in here. So here, one thing comes into play, that we do something which we call as an immunoprecipitation reaction. First of all, when you do this experiment, you add usually radioactive methionine or cysteine into the solution so that the proteins that are synthesized is radio-labeled. So they will have radioactivity. And on top of that, what you do that now, again, your primary secondary antibody comes into play, that you usually will have antibody against this protein. And they are usually immobilized in some agarose beads. So what you do that, you take the sample and put your antibody, primary antibody associated with the agarose bead there, and then you just mix them and then spin them down. And essentially what happens, that the agarose beads are heavy and they are going to pull down the protein. All other proteins or the junk proteins that you do not need remain in the supernatant. You just do this a couple of times. It will not be 100% purified or completely. But when you run the gel, those antibody and agarose beads will pull down your protein of interest. And as they are radioactive, this will be essentially put against an autoradiogram or a luminescent. At that time, it was only X-ray. Now you have light emitting chemicals as well. And you put an extra film in there, and you will going to see these bands, which are essentially this protein in presence and absence of microsome. So so the immunoprecipitation is just like, you can just Google it out how it is done. That is not needed for your exam. That is just if you're just interested that how the experiments is done, just without going in a shortcut. But the take home is that with microsome, you have a smaller protein, and the protein size appears to be larger in absence of the microsome. So This actually puzzled them, and they started to go into the detail of that. And what they found, that in the presence of microsome, the protein is essentially does have the signal peptide, and it is cleaved in presence of the microsome, which is that, and this is the protein which doesn't have the molecular weight of this. See that this is the intact protein with the signal. So what happened that right here, without the microsome, you do not have any translocation, you do not have the signal peptidase. So this is the protein plus the signal peptide. So the molecular weight of this is higher compared to this, where in presence of microsome, the signal peptidase cleaved the signal. So a couple of amino acids gone, and this is the correct size of the protein. And this is the first evidence that there was some signal that is involved in targeting these proteins to the ER. So after that, the important example also, just more detail about that, how you exactly know that where these proteins are, and you have two sets of experiment for them. So for both of them, we do a cell-free protein synthesis, which is another name of in vitro translation. In vitro translation, cell-free protein synthesis, the same. So the first one, what we are doing is like you take the ribosomes, mRNA, tRNAs, all the other things, and you synthesize the proteins in vitro, no microsomes present, just like the previous experiment. So this is minus microsome. But the only modification is that once you have completed the protein synthesis, then you add your microsomes. So this is adding your the microsomes post-translationally, once the translation is done. And you know that this time, that how much time will take, 5 minutes, 10 minutes, 30 minutes. After this is done, you add the microsome. Whereas when you do another set, what you do, that you have added the microsome at the same time, you add the reagents. So everything was present at the same time. And let's now take a look at what were the difference in between. So now what happens, that right here, what we was seeing, that there is no incorporation into the microsome and no removal of the signal sequence. And here the mature protein chain without the signal sequence. So there are two sets of experiment going on here. First, you take the two samples, and then remember that they can be radio labeled the same way, and you break this thing apart, do the immunoprecipitation, and run the gel just like this. So what you are going to see, that the one that you have added microsomes later after translation, the protein size should be like this. On the other hand, the one which you have added the microsome before starting the like all simultaneously, this is the correct size. So what you are essentially seeing, just see that right here, these are the one with the signal sequence, higher molecular weight. These are with the digested signal sequence, lower molecular weight. Remember that we are breaking this entire part out, then doing the immunoprecipitation, and then running the gel to see those. This is your first set of experiments. So this is higher molecular weight, lower molecular weight, or correct size. Correct size, larger size, or incorrect. Then the second question they asked is that, did these proteins enter into the lumen of the ER or not? That was the important one. Where they are they outside or are they inside? So what they did, that if they added protease to this experiment, proteins will be digested, degraded as they are outside on the reaction mixture. And if you add a protease to this experiment, protein will be protected by the microsomal membrane as they are inside. Proteases cannot cross the hydrophobic membrane. Remember that size is the first experiment where you are breaking up both of them doing the experiment. This is with another fraction where you have intact this and intact this. So do not confuse with that, the second one. If you break both of them apart and add the protease, protease will digest everything essentially. But here in this second fraction, you are taking this and taking these two separate tubes and adding protease. These will be digested, these will not. So essentially, if you run the gel, what you are going to see that minus microsomes, this will be essentially a black streak. There is nothing in there. You can see some black streak of amino acids at the very bottom, maybe. There is absolutely nothing in here. Everything is digested, but you will be able to see this protein because that is protected. And that gave the first impression that indeed the proteins which does have the signal sequences are extruded inside the microsomes. All right, there is a last set of experiment that you want to do. So the first one, difference in size. Second, without the microsomes added earlier, they are digestible. These are not digestible, so they assume that they are inside. And the third one they did, we are going to see that they added detergent to both of this, and then add the proteins, and everything is digested back again. So remember that detergent is going to make hole in this membrane, and now the proteases can go inside and will digest this as well. And that confirmed that they were indeed inside the membrane. They were protected by the membrane, but in presence of the detergent, they were digested. That will be essentially the next experiment. So this is essentially the experiment, that how we can tell the protein extruded into the microsome. So that's basically what we talked about. So this is just an idea that how we are taking a look. So here, take a look at this. So here we are looking at the ER, and the proteins are extruded into the lumen, and microsomes, these are attached to the ribosome. You just do the homogenization, and then you have two pathways, essentially. This is just the third set of experiment, which is an extension from the previous one. So if you add protease to these proteins where you have added, and this is what we call it as a co-translation of translation, essentially. So you have added your protease, all the proteases remain outside, and no digestion of the secretory protein, they remain undigested. But just only add the detergent, they are going to make the holes, detergents are going to be inside, you have to digest the secretory protein. So essentially resistant to protease and not resistant to protease in the presence of detergent, and become glycosylated by enzymes within the microsomes, essentially. So this portion we are not going to be interested in, that is not the question that we are asking. But just remember that these two are connected to experiments, that how it was understood that for targeting the proteins, you need a signal sequence. So all of them are essentially connected together. That you need a signal to be targeted into the ER, then the signal is actually cleaved, and the correct protein size is smaller than the signal protein. And during this process, the protein enters into the ER or the microsomes and remains protected by the membrane. And if you use detergent in that process, they will be able to digest. So that doubly confirms that they are inside. So definitely look at these experiments and make sure that you got this. Any questions on these? Good? Okay, all right. All right, so after that, some of the signal sequences, that what these signal sequences are, there could be stretch of a hydrophobic amino acid followed by some charged molecules. Remember that you do not do not have to memorize any of these signals. There are many different names that what you see that we call a signal peptide signal sequence. Sometimes we call it a leader sequence. We call it start transfer sequence. So many different names, and we'll explain that what those are. But again, memorization of this is absolutely not needed. So here you can see that the import into the ER, that's the top signal, retention of the lumen of the ER. So what we mean by that retention of the lumen of the ER, if a protein, as I said, that if this is a protein that works only in the ER, if you have these sequence, lysine, aspartate, glutamate, and leucine, this will keep the protein in the ER, and it will not make it transport it to the Golgi lysosome or the membrane. And you can see that mitochondria, nucleus, and peroxisome, these are their signals as well. So essentially, knowing one will tell you that how it is done. But in the mitochondria, it is much more complex because there it has to cross double membranes. Nuclear, it can go through the pore, but mitochondria, it cannot go through the pore. So it has to go through the double membrane, and there are two sets of proteins that actually does that. But again, detail of that is not needed. So now we are looking at just an overview of this, that how this ribosome, which is associated with an mRNA, is brought onto the ER, because the ribosome is in the cytoplasm. It gets the mRNA, and this is happening in the cytosol. no one knows that what mRNA it is reading. It reads the mRNA, and then the protein is coming out. So this is the mRNA. It's sliding or ratcheting through this, and this is your N-terminal signal. And again, as I said, that it could be internal as well. So usually this does have, that from the previous one, if you see that it does have a hydrophobic residue. You can see that right here, all the way that right here from leucine all the way this is the entire part. So it starts from here all the way this entire part is hydrophobic. So as soon as this protein actually comes out with a hydrophobic sequence, it is not favorable for those amino acids to be in the aqueous solution, which is the cytosol essentially. So what happens that there are some particles which are known as an SRP or signal recognition particle. These are made up of protein and RNA. There are plenty of these, and immediately they bind to that hydrophobic sequence and sequester. So immediately, as soon as that hydrophobic signal sequence comes out, so it just covers it out, wraps it around, so that hydrophobic sequences are safe. They are not safe means like carbon-chemically they are stable. Essentially, well, that's what I mean. Now, the interesting part is that the SRP also, when binds to this, it temporarily arrests the translation. Arrest means like it temporarily stops translation. So essentially what is happening, that as the ribosome is moving through, it is pushing the peptide out through this channel. Once the signal recognition particle bind, it stops that, and the ribosome is stalled in this place. It's not going anywhere. It is not done synthesizing the protein yet, but it just stops. At that point, what happens, that this signal recognition particle, it's just Brownian motion, just bouncing around, finds the SRP receptor, and it's an alpha beta subunit. This is on the ER membrane. So the ER membrane contains a SRP receptor, signal recognition particle receptor, and the SRP receptor finds its receptor and brings the translating ribosome onto the SRP receptor. Just remember that, the whole thing. Translating ribosome, protein coming out, the hydrophobic sequence recognized, signal sequence immediately recognized by the SRP, binds, stops translation momentarily, brings that whole ribosome along with the mRNA onto the SRP receptor. And then we're going to see that what's happening. This is just the overview of the structure of the SRP. Note that this is like detail of these things are not needed, and these numbers are not needed either. But just remember that it is a large molecule which contains this RNA. These are the RNA secondary structures and a lot of subunit of proteins, and it will have a hydrophobic area right here. See that right here? This is a hydrophobic area. Remember that it binds to the hydrophobic signal. So this is the area which interacts with each other and sequence and cover that hydrophobic area. So this is essentially the binding domain onto the signals, essentially. So several proteins, very large protein, and SRP, and it also needs essentially GTP to function. So here there are some of the homologs of those from different species. So this is a hydrophobic binding rules, and you can see this like a dimeric, and this is a homolog of SRP receptor, alpha subunit. You have this GTP, and we are going to see that this GTP is hydrolyzed while it transfers those to the translocon in a structure. So we are going to see that. So this is essentially GTP is required for the SRP function, and this is essentially the structure of it. Just an overview of that. Detail of the structure is not needed, but remember that it is the GTPase domain of the homolog of the P54 subunit, which is the one right here, which essentially binds to the hydrophobic residues of the signals. All right, so after that, we are looking at the first one, that how the overview of how a soluble protein is actually coming in and delivered into the ER lumen. So we stopped right here that the SRP brings the transloading ribosome onto the SRP receptor. This docking, see that right here, this docking of the SRP to the SRP receptor and transfer of the ribosome to the translocon requires the GTP hydrolysis. So from here, this SRP is going to hydrolyze its GTP. It does have a GTPase function. So it will hydrolyze the GTP, and that will release this ribosome, and it will transfer it next to the translogon, which is usually the same. So what is happening essentially, that these SRP receptor, SRP, and the ribosome, they are moving around, and they actually find a translogon close by. And once this GTP hydrolysis takes place, this entire structure is transferred, the ribosome and the mRNA is transferred on top of the translogon, and the SRP dissociate, it charged by the GTP again. So GDP and PI, and it's transferred. See that there is a gate that is closed in here, and now once the ribosome actually sit on the translogon, this is the transmembrane channel on the ER membrane, this channel is now open. So what happens, see that right here, this signal peptide is also recognized by the translogon, it binds in there, and the signal peptidase is going to come over and cleave that right here, and the protein, which is, it goes through this channel and delivered into the lumen, and it will fold up and it will become a mature soluble secretory ER luminal protein. Remember that, mature soluble secretory. The reason is that it is in the soluble form. It cannot be, even in the secretory, does not mean that it is needed to be secreted outside the cell. Secretory means that it is synthesized on the ER. That's, remember that, on the ER pathway. It can be a resident protein of ER, Golgi, lysosome, or secreted outside. So all of them are secretory proteins. So that is the whole mechanism. So how exactly some of the scientists actually found out that that the protein is going through this channel, or the translogon, into the ER. So this 661 alpha is a translogon from the east, the reverse east, Saccharomyces cerevisiae. So, and they isolated this, and they made an in vitro translocation experiment. So what they made, that they made an artificial mRNA about which can make essentially about 70 amino acids essentially. So this is a prolactin artificial mRNA that made. The unique thing that all you need to know, that the artificial mRNA did not have a stop codon. It did not have a stop codon at the end of the RNA. This whole structure, the ribosome structure and the produced tRNA, and this is not released. It will remain stuck. So essentially, they, by omitting the stop codon, they wanted the last tRNA that is being there is stuck, and to the tRNA, the entire protein is stuck. And what they did is that essentially in the right here, they had a particular residue, like a lysine residue, on the protein that they give as a tRNA, as an amino acid, which can be picked up by the tRNA. And that lysine residue can be essentially activated by a laser light. So if that lysine residue is activated, it will, by the laser light, it will cross-link itself with the corresponding protein, which is in the translogon. What it means, just think about this, that they assuming that this is what is going to happen, and there is a lysine residue in the protein in the middle of the translogon. So if this is the condition of the protein is hanging in there in an in vitro translation experiment, there is a lysine in the middle, and they modified this lysine with a light activated lysine. So if they shine a laser beam, it will cross-link it, essentially making a covalent bond with the translogon protein. So what does that mean? So after this experiment was done, they isolated the translogon using its antibody. So essentially you break up the everything, and if they see that when you isolate the translogon and run, it's like a 1200 kilodalton protein, very large, but if you just run this on a gel, if you see that along with your purification, you have isolated this cross-link peptide also, that means that this was indeed inside the translogon and cross-linked. If this is going somewhere else without this lumen, then it would have not cross-linked. If you isolate, break this entire structure and isolate just the 61 alpha translogon, you will only see the translogon. You will not see this protein. Only if this goes through the channel and if the lysine interacts with this light activated cross-linked ingredients, then they are co-unified. So this is how essentially. And now, so these were all experimental studies. So you can just take a textbook and read that in a little bit more detail. But more detail is not necessary. So if you are looking at the translocon, essentially, you're looking at the top, essentially, and this is a pore. And these are some of the isoleucines close to that, which essentially help in closing these. So there is a plug that can be done in here. On the other hand, that is one of the important things. Just remember that the translocon, it actually does have these N-terminal. It starts, see that right here, it goes all around and then it ends in here. But if you just look at the structure, that it just goes around like this, there is a gap in between. Remember that you have a string and you are looking at the top of the membrane. So suppose this is the membrane. So far we were looking at the translocon from the side. Now what we are looking at the translocon from the top. And from the top, think about that, you have an N-terminal, C-terminal, and there is a space in between these alpha helices. So what we are going to see that this is the pore through this, the protein is going to the lumen. But for the proteins which go into the membrane, the proteins can translocate on this side, or they can just come outside of the translocon and be inserted into the membrane. So we are going to see that the membrane-bound protein, how they actually operate. So here, we are still looking at a detailed picture of the same one. So this is a repeat picture of the same one, just a little bit complete. You have a transloading ribosome with an N-terminal signal sequence. This is caught by the SRP. SRP brings onto the SRP receptor, and see that this is a GTP. Remember that both of these GTPs are bound to the SRP, not the SRP receptor. It gives you a kind of a false impression that the GTP is on the SRP. Remember that both the GTPs are bound onto the, both the GTPs are on the, remember that right here. So there are two GTPs, and this is what is being shown, not on the SRP receptor sometimes. And then once it finds the translocon, see that the GDP is hydrolyzed and the SRP is again recycled back. And right here, the translocon is open and signal peptidase comes in, digests the signal, and this protein is extruded into the lumen of the ribosome. So this is essentially the mechanism of the soluble proteins. So just remember that. Now, after that, there are some exceptions that happen. That here, it's in the east, which is also period, the translation actually happened. The translocation actually happened post-translationally. So just remember that there are two words that we are juggling with. Remember that the translation is the process by which the polypeptide is made, and the translocation is the movement of the protein through the translocon into the membrane or the lumen of that. So just remember that translation, translocation. Remember that in protein synthesis, these terms, just make sure that you have clear understanding of that. So post-translational means that this protein is already synthesized in the cytosol, but it still contains a sequence. But it comes later and then inserted to the translocon and brought in here. So the ribosome doesn't sit onto the translocon compared like this in here. So this mechanism a little bit different. But there is a problem. What happens that in here, while translating, this protein will never be able to go outside once it's released, because this is essentially either closed or the ribosome is sitting in here. But there is a problem for these proteins if they're translocating post-translationally. This protein is translocating, and this can actually move outside backwards as well in the cytoplasm. So there is a mechanism. There is a protein called binding protein or BIT, and this binding protein actually attaches with a translocating protein. And see that this is essentially a protein which is beaded. So remember that your glucose comes in and it becomes glucose 6-phosphate so that it cannot leave. So this is the same way that BIT is coating this protein like these beads so that this protein cannot go backwards essentially. And then they are hydrolyzed and dissociate, and the protein is properly folded and remain. Once it's folded properly, it will not be straight polypeptide. It cannot go back once again. All right, so after that, this is all about your soluble protein. So if the secretory proteins, just remember that from the very first slide, if this protein is essentially this right here, soluble protein, then this is the process. Now what we are looking at, if the protein is inserted into the membrane like this or this or this, and they are membrane bound, how they are synthesized in the same pathway. So there are several types. So let's take a look at these. All right. So here you can see that these are what we call as a topology, and remember that word, we are going to talk about a lot about the topology of the membrane. So now you already know that this is your ER membrane, and this is the cytosolic side, and this is the lumen side, which is equivalent to the exterior. So this is also very important. Lumen is the exterior side. So here you have some of the single pass proteins. You can see that N-terminal in the lumen. This is N-terminal in the cytosol, N-terminal in the cytosol, C-terminal in the cytosol. Some of them have long N-terminals, long C-terminals, and all of these are possible. So these are the different topologies essentially. And here you are looking at a multi-class, and right here is a glued transporter. So you can see that 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. It's a 12-pass protein. And the N-terminal and the C-terminal are both on the cytosolic end. And we're going to see that. This is a very interesting thing that you're going to see, that if a multi-class protein does have the equal number of even number of passes, then the N and the C-terminal will end up in the same direction, either cytosol or in the lumen, but same. But if they have an odd number, so here is 1, 2, 3, 4, 5, 6, 7, the N and the C-terminal will be an opposite end. Now, this N-terminal doesn't have to be in the lumen. The N-terminal could be for a different protein. N-terminal could be at the cytosol, and the C-terminal could be here. But just the understanding that if they're even number, N, C-terminal are on the same side, either cytosol or exterior. If they're odd number, N and C-terminal are on the opposite sides or opposite faces, essentially. So now what we are looking at, that how these membrane proteins are essentially synthesized. For all proteins which are going into the lumen, the sequence or the signal sequence is always N-terminal. They are always N-terminal, but here there are just only one or maybe two proteins that what you are going to see. That's why I just did not cover that this one. Only one protein does have an N-terminal sequence. And remember that these are all membrane types, like membrane types protein. Like these are all inserted into the ER membrane. Again, they can be in the ER, they can go to the Golgi, lysosome, or the plasma membrane. These are the four destinations that they can be. So the first one, which is the type 1, and again, you do not have to memorize the examples of that. We are not going to ask that, but just all the types that what are their topology that you need to know. So the first one is exactly similar to the soluble protein. It does have a cleavable N-terminal sequence. Only the difference is that it is inserted into the membrane with a hydrophobic sequence, and it is not extruded into the loop. That's the only difference. On the other hand, see that the type 2, the N-terminal is in the cytosol, C-terminal is in here. Here the N-terminal is in the lumen, C-terminal is in the cytosol. Whereas the type 3, the most of the C-terminal is in the cytosol, very short N-terminal. This is essentially the opposite of that. This is what is called a tail-anchored protein. So there is a C-terminal, which is right here, and the N-terminal is mostly in the cytosol. This is a multi-pass protein, type 4, and there are two types, type 4A and a type 4B. We are going to see that, which are odd and even number. And then there is another one, which is a GPI-anchored protein that we are going to see. This GPI-anchored protein is actually synthesized like a type 1 protein, which does have an N-terminal sequence. And then the protein is cleaved from this luminal side and transported onto a lipid by an enzyme called transamidase. So we are going to see each one of them, that how they are essentially done. So right here is that this is the most similar to the ones which is in the lumen. So if you see that right here, again, remember that all of them will have the same picture of the SRP, arresting the translation, bringing it to the SRP receptor, handing it over to the translogon for this to happen. So what is happening that right here, this polypeptide chain is coming in, signal peptidase cleave that, but see that this is where it becomes different. After the protein is extruded for a while, there is another sequence comes in, and this is called a stop transfer anchor sequence. This is a hydrophobic sequence, and this is membrane spanning. So this is going to be inserted into the membrane. So when this hydrophobic sequence comes in, there are some hydrophobic sequences around that area. So essentially, if you have seen those before, this detail is not necessary. But what happens is that around this area, there are some hydrophobic sequences. So usually, most of the time, that when a protein is actually moving through, essentially, there are hydrophilic sequences. Hydrophobic sequences doesn't interact with them. So they are basically repelling it and allowing it to just pass through. So it is just intruded into the lumen. But when there is a hydrophobic sequence, they actually do interact with them because there are hydrophobic sequences of the translogon in here. And then it just arrests the translation for a little while. So that is also just like the ASAPI, and that's why we call it stop transfer anchor. So here it is arresting it, and there is a conformational change, and the translogon opens a lateral gate. So remember that it is like a clamp, and it just opens, and the protein actually moves from this inner wall into the membrane, if we're just looking from the top. So this is exactly what is happening. The protein is laterally exiting the translogon, and now the protein is in the membrane. And this is the hydrophobic sequence. This is the membrane spanning domain, or MSD, membrane spanning domain of the protein. And now it resumes the translation back again, and see that this portion of the C terminal, this is now translated by the ribosome, and this becomes the C terminal. So now you have an N-terminal protein, membrane protein, but you have a membrane protein where you have the N-terminal into the lumen or the C-terminal in the cytosol. If it remains like this here or in the Golgi or the lysosome, this will remain like this. But if this becomes exterior or the plasma membrane protein, this N-terminal will go on the outside of the cell, and this will remain in the C-terminal. So definitely remember that the topology of the membrane, that if the N-terminal of the ER, the N-terminal of the protein remains in the ER, and if that protein, this will be for your exam, if that protein becomes a plasma membrane protein, the N-terminal will become the ER luminal protein right here, that will become or face the exterior. So understand that mechanism. That's essentially what it is. All right, so now we are looking at a type 2. This is just type 1 in reverse. Does not have an N-terminal sequence, so this is a little bit different. So here, what you can see that there is only one hydrophobic sequence, and N-terminal is in here, C-terminal in here. So what is happening right here? So here, the story is a little bit different. The N-terminal right here, all the way up to these plus signs right here, all the way before this signal, this entire portion, it is synthesized in the cytosol. Remember that? There is no signal. There is no signal from here all the way up to this. That's why this is, the ribosome is hanging in the cytosol synthesized. Then you have the signal comes out. This is a signal anchor sequence. This signal, when it comes out of the ribosome, SRP binds to this, and then only at that time, it brings in the ribosome on top of the translogon. Always a very unique thing actually happens. Just before that signal anchor of the hydrophobic sequences, there are some strong plus charges in there. And at the mouth or the opening of the translogon, there are also plus charged amino acids. So this plus charged amino acids never enter into the lumen. So here, the protein is lowered into the lumen like a loop, like a U-shaped loop. Make sense? Like an U-shaped loop. Say that right here, this portion remains outside, and this portion is essential. So this is now moved in here. Remember that this red part was earlier here. red part was earlier here, so I can just draw this. Just makes sense a little bit better. So this red part were earlier like this. I'm sorry, well here, and then it moved in this direction. So this entire portion was lowered like a loop, and you can see that it's now being lowered as a loop as well. So that's essentially what it is. So now what happens, that the rest of the protein is inserted as the loop, like this U-shaped structure like here, and then finally when it is released from the ribosome, the entire C-terminal, which is coming from here, it falls back into the lumen. So the N-terminal actually remains in the cytosol, and this right here, the hydrophobic sequence, is going to move. So here there are a couple of things that is very important. There is a difference between the signal anchor sequence and right here, which is called a stop transfer anchor sequence. So signal anchor is what we call as an SA sequence, and STA is a stop transfer anchor sequence, STA and SA. What is the difference? STA actually appears as a transmembrane retention signal after a signal sequence. There is already a signal sequence. After a signal sequence, if there is a membrane retention signal, that means a membrane insertion hydrophobic sequence, and that stops or arrests transfer for transferring that, we call this a stop transfer sequence. It is not a signal. No SRP binds to the STA signal. Remember that stop transfer anchor signal, no SRP binds. SRP binds only the signal sequence. On the other hand, here, if we just go back to here, the type 2, right here, I'm just skipping those. This is the type 1, yeah, right here. All right, so on the other hand, right here, you can see there is no N-terminal sequence because N-terminal is in the cytosol, so there is no sequence. This is the only sequence which the signal as well as the membrane insertion signal. So that's why we call this a signals anchor sequence. So this is a signal, and so there is no other signal before that. That's why it is a signal. So SRP binds to this, bring it to the translocon, and the same signal acts as the membrane insertion signal because it's hydrophobic. So just remember that. What is the difference between these two? In the stop transfer anchor sequence, preceding this, there should be a signal that brings it, and this is just only helps in the transferring this portion into the membrane, and not in this one. All right, so after that, let's see that what is happening in the type 3. So the type 3 just have a tiny N-terminal sequence in here, and this is almost the same as well. So you can see that N-terminal enters in here. There is a hydrophobic sequence, but what is the difference? There is no N-terminal signal sequence. This is type 1, except the N-terminal signal sequence. So if this is the only sequence in here, so what will be this sequence? SA or STA? Is it a signal anchor sequence or stop transfer anchor sequence? What is that? Anyone? Louder? Signal, yeah. This is the only signal, so this will be a signal anchor sequence. See that there is no signal prior to this. So this is the only signal, that's why this will be a signal anchor sequence. Remember, type 1 already had a signal sequence here. So type 1, when this appeared, it becomes a stop transfer anchor sequence, but not a signal. Signal was already here, and this is the only one, so this is a signal anchor sequence or SA sequence. And as you can see that it is transferring into the part, and this plus area is essentially that will not be able to enter into the lumen, so this remains on the cytosolic side, essentially. So that's the important point. All right, so this one is important, the tail anchored protein. Here, there is an involvement of another set of proteins. And here, we call this as a GET complex. So this is equivalent to SRP, but there is a big difference in there. That's what we're going to see. So if a protein is tail anchored, which means the insertion of the protein is at a C-terminal. have a huge problem. The protein, the signal doesn't come out until the protein is completely synthesized because it's at the C terminal. So there is a problem. So remember that for the C terminal, and definitely write this one down, that for the tail anchored protein, the translocation is post-translational. For all of the other, that type 1, 2, 3, that's what you have seen, it is co-translational translocation. As the translation is happening, translocation is happening. Co-translational translation. Here, it is post-translational translocation. The entire protein is synthesized by the ribosome in the cytosol. The protein is released with a C-terminal hydrophobic sequence, which is very unstable. And what happens that immediately it is covered by these protein complexes, HGG2, GET4, GET5. That's a GET complex. These are SRP-like sequences, but a little bit different. But all their function is essentially to cover this C-terminal hydrophobic ends. And then these GET complexes get the protein into the GET3 complex. This is just like your SRP, but big difference, it works with ATP, not GTP. Absolutely an important question for your exam, that which of the following works without ATP, without GTP. So only the tail anchored protein synthesized by the GET3 gets with an ATP, the rest is all GTP. So the GET3 complex actually brings it to a receptor, GET1, GET2 receptor. This is just like an SRP alpha beta receptor, just a different name. ATP is hydrolyzed to ADP, and the protein is inserted into the translocon, just the same fashion. And the C-terminal essentially moves out into a membrane. So the only difference, it is a post-translational translocation. Entire protein synthesis happens in the cytosol. Then the GET complexes, with the help of ATP, brings it onto the translocon to insert it onto the membrane. Very different from the other ones. All right, so after that, what happens in the proteins, which are multi-pass? If you just understand one of them, you will just get all of them. So first, everything is the same. You get the SRP, and you get the first one. See that you do not have an N-terminal signal. So the first signal is the signal anchor sequence. So the red one is the signal anchor sequence, SA, and the next one will be stop transfer, STA. So what happens that after this signal, the ribosome is essentially not sitting onto the ribosome, but it's just remain hanging in the cytosol, because once this portion is essentially inserted, the ribosome is not going anywhere. And still the picture actually shows the ribosome is actually hanging on in the cytosol. This is actually wrong. The next edition of the book will come up. Now it is known that there is a translocon complex which actually keeps the ribosome pretty close to the membrane, but it's not in the textbook yet. But this has already been discovered in 2020, 2021. So more evidences are coming up. So essentially what you're seeing, that first is a signal sequence, then stop transfer anchor sequence. And then you have the next signal sequence comes up, and a SRP-like particle brings it onto here. So you have the next signal anchor sequence, next stop transfer anchor sequence. And then you will have the next signal sequence, next stop transfer anchor sequence. So each time, the signal anchor sequence is recognized by the SRP and brought into the translocon. And the other one is a stop transfer, just to transfer the next one into the membrane. So this is how your multi-domain proteins are essentially synthesized. So the next one is your GPI anchored protein. And this one is easy, which is not shown in here, the N-terminal signal sequence. This is essentially synthesized just like your type 1. It does have an N-terminal signal sequence, which is cleaved by the signal peptidase, and now it is inserted into the membrane with the C-terminal N. Now what happened, this protein is important, and this is very important for your exam. Know the GPI transamidase, it is not aminase. It's D, it's not N. So just remember that word, that usually people get confused. So just remember that. Once this protein is synthesized, the GPI transamidase cuts it and transferred it onto the lipid, which is the GPI anchor. And this is essentially degraded. So this is essentially the process. So a lot of people actually do ask is that why this is done in such an elaborate way, that bringing it in here, and why it is not inserted in here. So there is a little bit different. Remember that this is lipid anchored, not protein anchored essentially. This protein has a lot more ability to move around in a certain area, particularly lipid raft, compared to these proteins. So entirely different phenomenon. So only the N terminal is used, the C terminal is just only for the manufacturing process and then degraded essentially. So this is your GPI anchored proteins, how they are synthesized. All right. After that, what we are looking at, and this is some of the things that you can do a little bit of homework. We will start with, if we have time, we can do one iClicker. But the rest of them, we are going to do it in next class, so that you just prepare this. So essentially what you do, that take the type 1, 2, 3, 4 proteins and draw the linear structure of them. What do we mean by that? I'm sorry. So take all of these protein structures right here like this, and then draw that. If you just draw them right and left, linear fashion like this, how it's going to happen. So type 1, signal sequence, which is cleave, lumen, you have a 6 stop transfer anchor. That type 1 doesn't have an N terminal. This is a signal anchor. Remember that the plus is towards the cytosolic side. This is very important. Then you have the luminal side, plus is towards the cytosolic side. And now see type 4A and 4B. Idea, and put the 4 parts right here, and don't write which on the cytosol or which in the lumen. Just looking at the charge, you will be able to tell which part of the protein is in the cytosol, which part is in the lumen. Take a look at that. Right here, the plus side is on the cytosol. The plus side is in the cytosol. See right here, this will be a loop around the cytosol plus, but the plus side is in the cytosol, plus side is in the cytosol. And remember that what is happening here? First one is a signal anchor, then STA, then SA, STA. Here, you have an odd number. That's why you have first 2 signal sequences are anchored, because remember that the first one is inserted into the membrane, then this is in the cytosol, and the second signal need to bring it to the membrane, and then SA, STA, SA, STA, SA, STA varies. So this portion is exactly same as this, just you need another extra signal, because otherwise you will not be able to bring these 2 onto the membrane. Because this is once in the membrane, this is already associated with that, so it can go in easily. So the type 4A will have alternating SASTA, type 4B with the odd number will either, depending upon if the N terminal is in the lumen, it should have an extra SS signal at the luminal side. If the N terminal is on the cytosol, it will have an extra SS sequence at the C terminal. So just mathematically, draw these out, and you can just do that. And what we will do essentially, let's do one question, and we'll do the hydropathic plot in the next class. Just let's do, and this will not take more than a minute. So that's why this is super easy. The rest of them, we are going to do it in the next class. One minute, not more than that. 1:43, so we're good. That's all. 1 minute. Just make sure that read this material so that we will start with 3-4 iClicker question in the next class. So make sure that you get this one and also read the part 2. All right, stopping it. Last chance. That should be enough. All right. Good job. All right. All right. Have a good weekend, and we'll see you next week.